Hey class, it's Jamie Batts, your instructor for AMP2. We're going to move in through more of chapter 20, looking at the adaptive immune response and how these class 1 and class 2 MHC molecules play a role in the immune response. So, why am I just having trouble switching? There we go. That's where we left off. Let's talk about how the antigens are recognized. So that is the first thing that has to happen in order for anything else to happen for specific immunity. You have to recognize that the antigen is present. So an inactive T cell will bind to the MHC antigen complex. So backtrack a little bit. Class 1 and class 2 MHCs. Class 1 are your body cells. Class 2 are your immune system cells. They gobble up a virus or a bacteria, they produce that antigen on the outside, right? So a T cell has to recognize that, an inactive T cell, right? They're just floating around, boom, they bind. And then it's like all the magic starts to happen, like the lights turn on, so to speak. This inactive T cell on it has special receptors to recognize the class 1 or class 2 MHCs when they're bound. So they'll recognize, oh my gosh, this is a class 2 MHC, I'm going to bind to this. They will only bind to a complex that contains the antigen that the T cell is already programmed to detect. Remember, this is specific immunity here. So if the T cell doesn't recognize whatever that antigen is, it's not going to bind. It has to recognize that antigen. Hi, Tori. What do you need, sweetie? Okay, I'll get you something to eat in a few minutes. Okay? I'll be right up. So here's the picture of all the magic. You have your inactive T cell bumping into your infected body cell or antigen presenting cell, APC. And this is going to trigger this kind of receptor activation where you have your receptor and your MHC protein binding. There's also something called an antigen recognition protein that's on the T cell. This antigen recognition protein is going to play a role with the next couple of steps. So you had MHCs on your invaded molecules, right? Or on your invaded cells, on the body cells or on the white blood cells. On your T cells, you have CD markers. CD stands for cluster of differentiation. So there's a special marker or cluster of proteins, they're membrane proteins, that are on the outside of our T cells. They can be CD8 or CD4. CD8 markers are on CD8 T cells. They're just going to respond to class 1 MHCs. CD4s respond to class 2s. Make sense? They're just special recognition to say, oh, you know, you're my kind of MHC type of a thing. So you have CD8 and CD4 CD markers. The CD markers are the markers that are on the T cells. Let's talk about how CD8 T cells are activated. And again, this is a lot of words. We're going to draw it out. We'll map it out. It'll be fine. See if you can map it out while we're talking about it. That's even better. So first thing that has to happen, antigen recognition. So the CD8 T cell is going to encounter the class 1 MHC that has that specific antigen on it that it recognizes. Right? That's just a chance, right? It's by chance this happens. After it binds, like we saw in that picture a few slides ago, there's going to be co-stimulation happening. Co-stimulation is physical or chemical stimulation, basically like the safety on a gun. It's preventing that T cell from mistakenly attacking any of the normal cells. It's kind of like that T cell grabs on and says, all right, I think I recognize you. You're an invader. You're an invader, right? Yes, I'm an invader. Are you sure you're an invader? Yes, I'm an invader. Okay. That's the process of co-stimulation. It's this kind of double checking that happens just to make sure that the T cell is about to attack something that's actually an invader. So after it's been activated by that co-stimulation, there's 3 types of CD T cells that will be CD8 T cells that are going to be produced. They're all going to be sensitive to that same specific antigen that the T cell bound to in the first place. It's going to produce cytotoxic T cells, memory T cells, and suppressor T cells. So these three types of cells are going to be produced after this co-stimulation. Here's how it is. Antigen recognition, checking, co-stimulation. You cool? Yep, I'm going to attack you. Okay, go ahead and attack me because I don't belong here. And then we're activated. So let's look at the process of what they actually do. So the cytotoxic T cells, these are destroying the abnormal infected cells. They have to have that specific class 1 MHC, the specific antigen. They're going to release perforins and perforate the cell membrane like we talked about before. They can also activate the genes inside of the cell to target apoptosis or program cell death. They could also release a lymphotoxin, which is going to disrupt the metabolism of the cell. So depending on what the cell is, it may, or what the pathogen is rather, it may attack one of those three ways. So memory T cells, these are produced, they do not differentiate, they're just there after that first exposure, they're produced. And so your body will keep these kind of in the library arsenal, so to speak, and then when the second exposure comes, those memory cells can become cytotoxic T cells and attack. The suppressor T cells are going to limit the response of other T cells and B cells, basically putting up the police tape, so to speak. Hey guys, back up, we don't need you here, go home, there's nothing to see, type of a thing. So they're acting only after the first antigen exposure, after that first initial response, to clear the scene so that the cells can be destroyed appropriately. So there's where you see our cytotoxic T cells and what they're doing. There's three modes of activation: perforins, apoptosis, or lymphotoxin. Memory cells are produced, they're inactive, they wait around, and then suppressor T cells make sure no other T and B cells interfere. So let's go ahead and chart this out before we, and you can pause the video here, my typical spiel. You can pause the video here and answer these three questions. We're going to make a graphic organizer here of this immune response. Oh, my camera is like attached to this cord thing, and it makes it not want to stay in place. All right, so this is our immune response. So in a tissue cell, that's what we were just talking about, tissue cells. Tissue cells have a class 1 MHC created. A class 1 MHC is created. Class 1 MHCs bind to CD8 T cells. When that CD8 T cell binds, we're going to have this co-stimulation. You cool? I'm cool. I'm going to attack you. Okay? Yep. Go ahead and attack me. That whole thing. Co-stimulation. Co-stimulation is going to make the cell undergo mitosis. From mitosis, you have memory and cytotoxic T cells produced. And the T cell, the cytotoxic T cells, these guys wait, inactive. Right? Our cytotoxic T cells are going to destroy, target, destroy target cell. Three ways, right? They have the perforins, they have apoptosis, or they will stimulate apoptosis, and they have the lymphotoxin to interfere with the cellular metabolism. Right, that's the whole process. So that's what they CD8 T cell. Let's look at CD4 T cells. Go back to screen share mode. Why don't I recognize which? I think it's this one. My computer's being weird. All right, so CD4 T cells. These have to be, these are going to respond to a class 2 MHC. So what we're just talking about is in a normal body cell. What we're going to talk about now is an immune system cell that has been invaded by a virus or that has engulfed a bacteria. Co-stimulation occurs again. That's completing the activation process. And then there's a series of divisions. You have daughter cells that are going to differentiate into helper T cells. Remember before we had memory and cytotoxic? Here we have helper and memory cells. So these helper cells secrete cytokines. Cytokines are going to activate cytotoxic T cells and B cells. So they're going to activate the cell-mediated and antibody-mediated defenses. So CD4 cell binding to a class 2 MHC, and that is going to, of course, produce this co-stimulation. You cool, I'm cool. Okay, I'm going to destroy you. After that, our T cell, our CD4 T cell differentiates into a memory cell and a helper cell. So our helper T cells are stimulating B cells and other T cells, and we'll look at that in a second. So in order for B cells to become activated, they have to become sensitized. This sensitization is when antigens are brought in to the B cell through endocytosis. They're placed on the outer surface and of the class 2 MHC. So that is then going to then require the assistance of a helper T cell. Helper T cells will then become activated, and they'll bind to that MHC complex of the B cell and produce those cytokines to help get that B cell activated. There it is. Here's our inactive B cell. Those antigens bind to the surface of our B cell, and then our sensitized B cell has the antigens bound to it. And here's the whole process. So you have your class 2 MHC bound by the CD4 T cell, co-stimulation occurring, those cytokines are being released between the T cell and the B cell. So the CD4 T cell, which has already bound to the class 2 MHC, is going to also bind to a B cell. This B cell produces another class 2 MHC and is going to present this antigen on its surface. So now our B cell is going to divide. The B cell is going to do the majority of the work in the case of CD4 T cell and B cell activation. So when we're looking at the main mechanisms here are CD8 T cells and class 1 MHCs. Those are mostly, those are our body cells. Those are mostly, all just cytotoxic T cells doing the dirty work. When we're talking about our immune system being affected, our phagocytes, like our monocytes or other lymphocytes being affected, our CD4 T cells and B cell activation for class 2 MHCs, we're looking mostly at an antibody-mediated defense that is supported by T cells. If that makes sense. So stimulation by cytokines is going to cause a series of cell divisions. Our B cells are going to divide. So that B cell that was initially bound to our T cell there, our helper T cell, is going to undergo mitosis. It's going to produce a memory B cell and a plasma cell. The memory B cells do the same thing the memory T cells do. They hold around, they wait around until they need to be activated after a second exposure. The plasma cells are going to differentiate and secrete 100 million antibody molecules per hour. So the plasma cells make the antibodies that your body needs in order for agglutination to occur. Here's the process. So you have those helper T cells releasing the cytokines, activating the B cells. The B cells are going to divide into a memory B cell, which waits, an active B cell, which differentiates into plasma cells, and the plasma cells differentiate into antibodies, or produce antibodies, I should say. Here are the questions for this little section. While you're gazing over those, I'm going to erase. Where'd my husband's hat go? It's black. You won't even notice. I'm going to erase this and draw out the other half of that graphic organizer. Now, I would, if I were you, do this all on the same graphic organizer that we just had. For my purposes, I have this super small whiteboard, so I can't do that. But this is the second half of that same thing. So at the top, we had immune response. All right, so here we're looking at a phagocytic cell. Sorry, Lily. It's my dog. I just stepped on her. So here we have a phagocytic cell. Phagocytic cell produces, I gotta write smaller. There's a lot on this one. Produces class 2 MHC. Our CD4 T cell binds. Then we get co-stimulation. You cool, I'm cool, okay, I'm gonna kill you, okay. That co-stimulation produces T helper cells and memory cells. Memory T cells. Memory T cells wait for second exposure. Right. T helper cells are going to bind to a B cell. Right. Remember, it releases cytokines. We'll just write that here. Releases cytokines. Cytokines are those little cell-to-cell messengers, right? So T helper cells release cytokines. They're going to bind to B cells. Those B cells will become sensitized. Right? Basically what that means is they're going to, B cells ingest the antigen and that's what sensitized mean. They basically ingest the antigen and they will become activated B cells. And they divide into memory B cells and plasma cells. Plasma cells produce antibodies. Antibodies will agglutinate. Memory B cells wait for second exposure. Ta-da! CD4. CD4 T cells on the class 2 MHCs. So let's stop this little video here, and then in the next video we'll talk about antibodies. Yay!